Okay, chapter 12, part 2. So we were talking about how we could take these fragments. You could take the same restriction enzyme and you can chop up the DNA and you would get different lengths. Well, how does that work? It's kind of hard to describe it. If you have a picture's worth 1,000 words. So here you go. You have two people's DNA. You got someone from a crime scene, and then you have the quite clearly innocent suspect. You take the same piece of DNA, so whatever you're looking at, chromosome 5, lower section, blah, 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 wherever you're looking, and you cut it with the same restriction enzyme. And this one cuts between, the C, G, whatever. It's going to cut in the same spot, right? So this restriction enzyme cuts here. And down here you can see, okay, they have the same sequence in the same spot. Maybe that, area codes for something we all need or something. But this person, they have another CCGG sequence here. So their DNA gets cut 2 times. They get a piece up here, they get a piece in the middle, and they get a piece down here. The suspect, their DNA only has one of these sequences, and this one cuts 1 time, leaving 1 big piece and 1 little piece. Remember I said when we look at forensics, it can be up to a 1 in a billion chance of messing this up. Well, and I said it depends, right? Well, it depends on which restriction enzyme you use. If you use one where there's like commonly a bunch of similar sites, you're not going to get as good results, but they figured out which sites tend to vary and are rare, and they use those for more accuracy. Okay, and then you run those people's DNA samples, because you can't see that it was cut into 3 pieces or 2. How do you know one was cut into 3 pieces or into 2 pieces? Well, here's the one that was cut into 3 pieces right here. You got up here the longest piece, you got the middle longest piece, and then you have the shortest piece. And this person just had the 2 pieces, right? So you can see now quite clearly that is not the same DNA, right? If it's cut in 2 different spots, that is not going to be the same DNA. All right, now remember these probes. These probes can be radioactive or fluorescent, and it's going to attach usually to the complementary strand of DNA we've created, and it's going to bind to it, and then we know, because you can't look at DNA. It's either got to glow or you got to make it radioactive or something. And we'll know if they attach that is the strand we are looking for. Okay. So let's say you're looking for, I don't know, cystic fibrosis or some genetic disease, and you find someone who's definitely a carrier. What you can do is you can run their DNA, and you're going to get just a bunch of DNA. If you use a restriction enzyme, you're just going to get way too many. So you take the paper, you literally take blotting paper. Go ahead. You can literally take blotting paper, blot it, and then, so now it's going to have all that DNA on this paper, right? All these lines are going to be on this paper here. So here you go. These lines are going to transfer to this blotting paper. Then you can take this radioactive probe of the strands we're looking for, the ones we know code for cystic fibrosis, because who wants to sort through, a cajillion strands of DNA? And it will attach just to the ones we're looking for, whatever gene that is. And then we can compare the film. You literally, with radiation, you can literally expose film using the radiation and then find just the genes you're looking for. All right, you guys are way too young to remember that DNA technology was like a big deal when they finally let it into courts of law. People were like, oh, no, that can't be accurate. No way. And now it's commonly used in courts of law, and people pretty much accept it. So, similar to finding carriers and other things, you can run this gel electrophoresis. You can take examples of, here you got the defendant's blood, you've got blood from, the defendant's clothes, which would be the crime scene, and the victim's blood, and wow, you can find there's a match. It's got both, his own blood and the victim's blood or whatever on it. And that's usually pretty definitive. You can run a couple different restriction enzymes and make it more accurate depending on what you're doing. And then gene therapy. Now, gene therapy, okay, is very, very exciting. It is not as easy as it sounds, because to get the genes to the cells that make new cells, you're talking like bone marrow cells for humans, or, ideally, if we knew when we were just fertilizing the egg, we could repair the DNA right there. But you never know until you have a complete human. And then the problem is, whatever gene you stick into, like the blood, isn't going to go and be reproduced in the brain. You stick it in the skin, it's not going to reproduce, anywhere else. Our cells by now have all the DNA in them, and they've all reproduced, and they've all differentiated. So it's a lovely thought. It's actually way harder than you think. So the idea with bone marrow therapy is that you can inject, because bone marrow is sort of like stem cells, it's going to make the blood cells. If you could get the DNA into the bone marrow, that would be fantastic. The problem is, I think they did this on some kids, and they actually triggered leukemia in a couple of them. And the reason is of course, the leukemia gene that's going to be active in the bone marrow, they think they got too close. I don't know how you can tell you're too close, but then you ended up triggering other problems, and leukemia was sort of a big problem, right? It's so not as easy as it sounds, but same basic idea of inserting. If you have a defective gene, the idea is if you have a defective gene, you get the normal version of the gene, you cut the defective gene, you insert it into the cells that are going to be the cells that are reproducing, and hopefully their descendant cells, their daughter cells, will have the new gene. And it says, unfortunately, progress is slow. That's because we're complicated animals. If we were like yeast, it'd be really, really fast. But still hopeful that there are genetic disorders that we can treat. Certainly we can make the protein, right? We talked about if you have a defective gene that's not making the protein, we can synthesize that and you can take it. The pharmaceutical companies are happy. But we'd like to fix the person themselves, their genes inside their body. Much, much harder than just synthesizing the protein in bacteria. Polymerase chain reaction. This is key. It sounds simple, but let's say you get some blood at a crime scene and you get a little tiny bit of blood. You don't have enough DNA to run the samples you need. Polymerase chain reaction, PCR, is just used to replicate DNA, and it just replicates it over and over and over again, and they can take one piece of DNA and make it into thousands in a couple hours or something. So before PCR, we couldn't really do a lot of genetic things because you didn't have enough DNA to work with. The cool thing about PCR is it just uses, it's a simple idea. The DNA, you're going to heat it and then cool it, and when you heat it, it's going to break apart into two strands, and then you use DNA polymerase to make a second strand. Sounds easy. The problem they ran into is every time you heated it, you were killing the DNA polymerase, and they finally found some DNA polymerase that was heat tolerant, came from like a bacteria by heat, these water vents in the ocean or someplace really hot. I can't remember where, but it was, some really hot place, and that DNA polymerase was good to go and could withstand the temperatures of PCR. The Human Genome Project, pretty much done. By now, it was begun in 1990, and what they wanted to do was physically map the human genome, just like it says. And computers are excellent at this. And there's different ways of doing it. You can kind of chop up the DNA and have computers piece the things together, but it has taken off. There's a couple companies that were working on it. And what you get is sort of insight into humans. And the pharmaceutical companies would love to use it. They're working on sort of DNA specific, so drugs, like drugs specific to your DNA. What it really helped too with was evolution, because they're mapping not just human genome, but like amphibian, like frogs, mosquitoes, different genomes, and then we can compare how far apart they are and kind of do an evolutionary tree of how far apart the genes are, how far back in millions of years would you have to go so that each species diverged from some common ancestor. Okay. Most of the human genome, 95%, 90, I don't know what it is, it's a large percentage, 97% doesn't even contain genes. And for a long time they're like, oh, it's junk DNA, we don't know what it does. Well, it turns out it does a lot of things. We still don't know some of the stuff it does. There's stuff that is preserved in our genome that we don't know what it does. Some of it's very well junk DNA, replicated DNA, maybe. But we shouldn't call it that until we know, I think. Just because we don't know what it does doesn't mean it doesn't do something. So, if we look at the haploid genome, so just a set from your mom, not doubling it, you're only talking 25,000 genes. I think before they used to say it was like, oh, it's going to be 100,000 genes. Humans are so complicated. Turns out there's only about 25,000 genes. Those are things that are code for proteins. And then there's a ton of what's called non-coding DNA. Again, this does stuff. There's structural stuff it does. Some of it we haven't discovered yet, but it's definitely not junk DNA. Much of the non-coding DNA consists of repetitive nucleotide sequences. So we have some repetitive ones called telomeres on the ends, but you also have these repetitive sequences that in the middle of them, you're going to have something called a transposon. And it's a gene that jumps spots. And the way it jumps spots, kind of think of sticky ends again, right? If you have this repetitive set on the left side and a repetitive set on the right side, and you have that all over the genome, then that gene can find a sticky end and attach itself somewhere in the genome, especially during crossing over, right? These things can move around. That's kind of cool. Do we know all the advantages? Yeah, no. Okay, so the science of genomics compares whole genomes, so the whole sequence of DNA. And usually we're talking here eukaryotic cells, but prokaryotic cells are easy to do, and so they've been done too, mostly because they're really easy, but things like E. coli have done. And of the eukaryotic genomes, you have things like Saccharomyces cerevisiae. Different ones have been done besides human because either genetics has an interest in them or industry has an interest in them. So some of the genomes that have been sequenced that aren't human are listed here. You have Saccharomyces cerevisiae, that's the yeast. You have E. coli, the bacteria that's in our gut. You have C. elegans, and these are, C. elegans is huge in genetics. They're easy to work with. Yes, the nematode is not very interesting, but it's interesting in genetics because it has a quick lifespan and you can manipulate it fairly easily. Same thing with Drosophila, right? Drosophila, fruit fly. Genetics, we love these things. They replicate very rapidly, so you can see instantly what you're doing, or I shouldn't say instantly. Within a couple weeks, you'll have a couple generations, and you can see genetics quite easily in the fruit fly. The mouse, mustard plant, rice, and of course humans. We have an interest in humans. So we use these for different reasons, but they all tell us something, and they tell us something evolutionarily as well as individually for each animal being sequenced. There's also sort of an evolutionary tree being made here. Now, proteomics is rise of the protists. Remember those guys who thought a long time ago that it must be proteins that were the genetic material and not DNA? Well, proteins are still very, very important, and so there is a way of mapping proteins, and it's kind of cool. It's similar to genetics because obviously the DNA makes the RNA makes the protein, but because of different ways of splicing things, introns and exons and such, the proteins can vary, and so it goes with it, but it's a kind of cool subset. Okay, so genetically modified organisms are transforming agriculture. Yes, they are. I think it's like 90% of the soybean oil you guys suck down is genetically modified. I can't remember all the percentages, but it's way up there. So if you don't think you're eating the stuff, you definitely are. I shouldn't say definitely. You most likely are eating genetically modified things. Plants are very easy to modify. Very, very easy to modify. Most of it is modified. Okay, so recombinant DNA technology. Yes, this looks similar to what we do into a plasmid, right, into the bacteria, but it's a little different in plants. It's not much different. You can use recombinant DNA to literally stick into plants traits you are looking for. So you can take the DNA, you can take it, you can put it in a bacteria, you can take it out using the same restriction enzymes. You can use the same exact idea that we do with the plasmid, but instead of that, you stick it into a plant cell. And the beautiful thing about plant cells is you can get this kind of glob of plant cells, and you can treat part of it with like root stimulating hormones, and you can get roots out of it, and you can get different parts of the plants by just applying different hormones to it. It doesn't work like that with people. It would be nice and easy if it did, but it doesn't. But it works great in plants. You can stick it into one cell, and you can literally, from this one cell, produce an entire plant, and that plant will have the new trait in all of its cells, which is the key part. It's not just in one cell. It's like we've used this almost as a seed cell, and we've made an entire plant out of it. A little different than a seed, though. We don't plant it. We're actually going to put hormones onto different parts and stimulate the growth in different ways. But still a cool way of getting the DNA into every cell in a plant. Okay. If you stick DNA from a different animal into an animal or a different plant into a plant, it is now called transgenic. So if you stick, a jellyfish gene into a tomato plant, you have a transgenic organism. If you just manipulate the DNA within the same organism, it's just genetically modified. So like the first tomato plants that were genetically modified, they flipped over an ethylene gene, and they just flipped it. They made it backwards so it wasn't working. That's not transgenic, that's genetically modified. But now we're talking transgenic, we're going to have genes from other organisms stuck into them. Okay. So there are, so many important crops and plants that are genetically modified. More than you want to know. So big question, can genetically modified organisms harm human health or the environment? And we try to be good in this country. I'd say other countries aren't as good as us. But yeah, the thing with plants is their genes can actually jump from the plant you stick it into the plants in the neighboring field. The pollen that carries the genes, there's actually a way that it's weird. It's not normal like what we think of reproduction in plants. But those genes have been known to jump. So when they make Roundup Ready, and they do make Roundup Ready plants, what that means is those plants are Roundup resistant. And the idea with genetically modified organisms, everyone thought, well, we'll spray less chemicals. What it turns out is we spray often more chemicals because we spray the whole field, and then the plants we have modified don't die. They are Roundup ready. The problem with that particular gene is it's been known to jump into other plants, and then you go use your Roundup in your weeds in your property, and they don't die, and you're like, why is that weed not dying? Well, if you're next to one of these fields, these genes can jump. Sort of a problem. That's the ecological damage I was just talking about from genetically modified crops. There's other things we do, make things grow bigger, faster, and you don't want those in your weeds either. So the problem with plant genetics is it gets a little messy. Things tend to move beyond your fences. Okay. So there's a genomics researcher, Eric Lander, who, it's at the end of the chapter there, that discusses the Human Genome Project and sort of the cool things that we're hoping will come from it. So the science of genomics compares whole genomes, sequence of many, blah, blah, blah. Right? And so when we look at what's going to come from the Human Genome Project, we find things like, well, some of our DNA is the same as mouse DNA and it hasn't really changed at all in millions of years. Even if it doesn't code for a protein, it must be kind of important